Hello dears, hello grade 9 students. Today we will talk about cell division, Unit 4, Lesson 1. Cell division, mainly we have two types of living organisms. As you know, we have prokaryotes and eukaryotes. Prokaryotes are the cells or the living organisms which have only one cell and the DNA is distributed randomly in the cytoplasm with no nuclear membrane, with no nucleus. While the eukaryotes, this is the living organism which have the nucleus and nuclear membrane and the genetic material incorporated inside the nucleus. Here is also another division for the living organisms. This is the division in which we depend upon the number of cells which form the body of the living organism, like as you see, unicellular organisms and multicellular organisms. The unicellular organisms, this is the organism which its body consists of one cell, only one cell. And the multicellular organisms, their bodies consist of many types of cells, many cells. We can join the two divisions together, as we see the prokaryotes as only unicellular organisms and the eukaryotes, some of them are unicellular and most of them are multicellular organisms. The multicellular organisms, like plants, animals, human, have two types of cells, somatic cells and reproductive cells. The somatic cells, they are divided by mitosis, which lead to the growth of living organisms. Somatic cells like stomach cells, skin cells, liver cells, these cells divided mainly by mitosis to give another cell have the same and identical to the original one, have the same properties and identical to the original one. And the reproductive cells, which are gonads, ovary and testes, which divided by meiosis and give sperms and ovum, gametes, sperms and ovum. So these are the two types, somatic cells and reproductive cells. Firstly, we will talk about the cell division in general. The cell division, mainly, we have the nucleus here in the cell. Here is the nucleus. Include the genetic material inside it. This genetic material mainly consists of chromosomes, which is responsible for the cell division. The cell division here have some aims, like in the somatic cells to produce another cell of the same and identical, of the same properties and identical to it, and in the reproductive cells to produce cells of sperms and ovum. So the cell division here is important for the living organism. The chromosomes are responsible for the cell division. Structure of chromosome, it consists of two connected threads at the centromere point. As you see here, two threads, 1, 2, connected in a central point which is known as centromere. The thread-like body is known as chromatid and chromatid. So the chromosome here is chromosome dichromatid, connected in the center of the chromatid in a point which is called centromere. The chromosome consists mainly of DNA and protein. This chromatid consists of DNA folded on protein portions, which is known as histone protein. Now we will study the two types of cell division. Firstly, we will study mitosis. Mitosis takes place in somatic cells like liver cells, stomach cells, skin cells, as we say before. What is the importance of mitosis? Mitosis mainly is important for the growth and compensation of the damaged cells. The growth of cells and the compensation of damaged cells mainly depend upon mitosis. Mitosis here, one cell give two cells. One cell give two cells, have the same properties and identical to it. Before starting the cell division, before starting the mitosis especially, the cell passes through some important biological processes. The interphase, this interphase in which the DNA duplicated and the content of the cell duplicated to laterally divided into two cells have the same content of DNA and the same content of organelles. So the interphase, it is the phase which precedes the mitosis. Phases of mitosis. Mitosis depend upon four phases: prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Firstly, the first phase is prophase. In this phase, the cell which contains the nucleus and contains the chromosome inside the nucleus, the nuclear membrane disappears firstly, and the genetic material comes to the cytoplasm to be prepared to enter the mitosis, and the two centrioles here in the cell, each one go to one pole of the cell, and the spindle fiber begin to extend through cytoplasm and attach to the centromere points of the connection of the two chromatids in each chromosome. Again, the prophase 1, it is the first stage of mitosis, the first phase of mitosis. Firstly, the nuclear membrane disappears and the genetic material comes to the cytoplasm. The two centrioles, each one go to one pole of the cell, and the spindle fiber spreaded to attach the centromeres, which is the connection point of the two chromatids in each chromosome. This connection and attachment of spindle fiber to the centromere is the first step to split the two chromatids from each other to do the cell division. Here is the prophase end with the chromosomes attached to the spindle fiber and centrioles from the centromere point. Second phase of mitosis is metaphase. Metaphase, here is the chromosomes now is found in the cytoplasm and arranged on the cell equator, in the middle of the cell, especially in the middle line, the middle imaginary line, which is cell equator. Here is the spindle fibers attached to the centromeres and be ready now to split in the third stage, which is anaphase. The anaphase stage, here is the spindle fibers attached to the centromeres and splits lengthwise by shrinking the spindle fibers and grasping the centromeres to be splitted, and the two chromatids, as you see here in this stage, separate from each other. Each chromosome separated into two chromatids, one to the pole and one to the other pole. Here is the anaphase process. A constriction will begin to appear here to be ready to separate the two cells from each other. The last phase is the telophase. Here is the spindle fiber shrink and disappear, and a new nuclear membrane is formed around the chromatids or chromosomes, which separate to each cell to form a new nucleus, special for each one of these cells. And this constriction increased till it separate completely to form two cells, as you see, to form two cells. Each cell have the nucleus, its nucleus and its DNA and cytoplasmic organelles, complete splitting here to the two cells from only one cell firstly, which we begin with in the mitosis or before the mitosis. Again, we can review the 4 stages or 4 phases again to show the mitosis phases. Number 1, prophase. Here is the nuclear membrane disappear, the chromosomes comes to the cytoplasm, and the centrioles go to each pole of the cell. The spindle fibers spread to attach to the centromeres, the connection point between the 2 chromatids in each chromosome. Number 2, metaphase. Chromosomes arranged on the cell equator and spindle fiber attached to centromeres. Number 3, anaphase. This anaphase is the splitting of the 2 chromatids and the centromeres lengthwise to form 2 sides. Here is the side of the newly formed cell and newly formed cell, chromatids here and chromatids here. Number 4, the telophase, which is the splitting of the 2 cells and the formation of new nuclear membrane to form a nucleus again and to separate 2 cells and producing 2 cells from only 1 cell. Secondly, we will talk about meiosis. The reproductive cells, gonads, testes, and ovary make this meiosis to form the gametes. What is gamete? The gamete is the sperm and ovum, which is the male sex cell and the female sex cell, which will combine together to form the zygote or the embryo in future. So here is in testes and ovary the meiosis done. Meiosis takes place in the reproductive cells, testes and ovary, and the importance of meiosis in the cell division is to form the gametes. These gametes have half number of chromosomes, which is, we can talk about human being, 46 chromosomes in each cell of the somatic cells and the reproductive cells, ovary and testes. But in the sperms and ovum, the number of chromosomes, half the number, 23 chromosomes only. Why is that? This is for being the after fertilization, the sperm and ovum will combine together to form again 46 chromosomes, which is the normal number of chromosomes in human. So the importance of meiosis is to form gametes which have half number of chromosomes to achieve sexual reproduction. Male gamete is called sperm, and the female gamete is called ova in the human. While in plant like this, the male gamete is called pollen grain and the female gamete is called ova also. What about phases of meiosis? Meiosis produces cells with half number of chromosomes of the parent cell. We can conclude that firstly, the original cell will give 4 cells, not 2 as mitosis, but 4 cells here. The original cell is 2n or diploid number of chromosomes, as I say in human 46 chromosomes, will give us 4 cells of 23 chromosomes, half the number, haploid number of chromosomes. The stages here begin with interphase as in mitosis, which precede the 4 stages of mitosis. Here is also precede the stages of meiosis. So the duplication of DNA comes through, and now we are ready to begin the meiosis. Meiosis is divided into 2 stages, meiosis 1 and meiosis 2. Meiosis 1 have prophase 1, metaphase 1, anaphase 1, and telophase 1. Meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. Firstly, meiosis 1, prophase 1. Here is the DNA is condensed here in the form of chromatin. The chromatin intensifies in the form of distinct chromosomes, comes through to be chromosomes. Nuclear membrane will be disappear, and the chromosomes comes to the cytoplasm here, freely in the cytoplasm. The 2 centrioles in the 2 poles of the cell, and the spindle fiber spread to attach the chromosomes as the centromere. In this stage, a very, very important phenomenon takes place. At the end of prophase 1, here is the crossover phenomenon comes through or takes place. The chromosomes here become as or arranged as tetrad, two chromosomes corresponding to each other, two chromosomes in front of each other. This is formed tetrad. The internal chromosomes here exchange some genes or some parts of the internal chromatids to form a new combination or a new structure for the chromosome. This is known as the crossing over phenomenon. This phenomenon takes place at the end of prophase 1, when some parts of the two inner chromatids of each tetrad, two chromosomes corresponding to each other, are exchanged to produce new genetic arrangements. This is make a genetic variation which will be appear in the next generations. So prophase 1, at the end of prophase 1, the crossing over phenomenon occurs. What is the importance of this phenomenon? It works on the variation of the genetic traits among the members. Here is the exchange occur here in the internal chromatids to form a new genetic structure for each chromosome, which will be transferred to the gametes and then to the new offspring to variate the traits or the genetic traits from individual to other. So it share or act as a source of genetic variation here. What are the phases of meiosis? Number 2, metaphase 1. We talk about prophase 1. At the end of prophase 1, we show the crossing over, and here is the metaphase 1. The chromosomes arranged here on the cell equator, but each chromosome corresponding to each other, as I say, and exchange occur in the prophase 1. Here is the metaphase 1, the spindle fibers attached to the centromeres and be ready to split in the anaphase 1. Every two homologous chromosomes separate to one pool. Here is the chromosome as a whole, separate to one pool, not chromatid as mitosis, but chromosome as a whole separate to one pool here and another one to one pool. So here is the formation of two cells from only one cell. Each one have half the number of chromosomes, 1n, only half the number of chromosomes. Telophase 1 is the stage of splitting the cell to two cells. The end result of meiosis 1 here is to form two cells, half the number of the chromosomes. Here is the phases of second meiotic division or the meiosis 2. The meiosis 2 actually we can consider it as mitosis, the same as mitosis. 4 stage, prophase, metaphase, anaphase, telophase, the same steps as in mitosis. Here is the chromosomes found in cytoplasm in prophase 1 and prophase 2, and in metaphase 2 the chromosomes arranged on the cell equator of the cell. Then in anaphase 2 the separation of chromatids, single chromatids to each pool of the two pools of the cell. Then the constriction occur, and the telophase is the splitting of the two cells which have half the number here. Each cell comes to give two cells here and two cells here, as I reach in the last step of meiosis 1. Two cells, each cell give us the separation here and give two cells half the number of chromatids, half the number of chromosomes, which is, as we say in human, 46 chromosomes give us 4 cells of 23 chromosomes, half the number, or the haploid number. We can say about the somatic and reproductive cell have diploid number of chromosomes, and the sperms, pollen grains, and ova contain half the number of chromosomes, which is 1n haploid number of chromosomes. Now, dears, we talk about cell division. Please contact us for any questions. Thanks a lot.